Today we will discuss a new topic about Calnexin, and that is how Calnexin helps in maturation and deacylation of these GPI-APs. The GPI-APs are expressed on the cell membrane, and we have understood so far from our previous videos that Calnexin and calreticulum cycle helps in properly folding of the glycoproteins that are expressed in the endoplasmic reticulum. So that was about the general overview about Calnexin and calreticulum cycle. So in this video we will discuss specifically about these GPI-APs, how this Calnexin helps in the maturation and deacylation of these GPI-APs. So in the case of this normal condition, what will happen is that the translation of the amino acids occurs in the endoplasmic reticulum, and then it interacts with this MOX GANAP, so that this carbohydrate structures, that is glucose 3 mannose 9 and N-acetyl glucosamine 2 will get uh, will get trimmed by this MOX and GANAP, and then remains this glucose 1 mannose 9 and N-acetyl glucosamine 2, which interact with this GPI-AP, and after that it is able to interact with this Calnexin, which is formed bond with this PGAPs. It helps in the deacylation. At the same time, it will increase the retention time of this newly translated GPI-APs, and by the time these GPI-APs acquired their native structures, they will then move towards this ER exit site with the help of this cargo P24 that will help in the transport of this lipid, that is GPI-APs, on the plasma membrane. And this PGA1 Calnexin, apart from this PGA1 Calnexin complex, this PGAP5 also helps in the deacylation of the GPI-APs. And if the cell line does not have both the Calnexin and calreticulum, what will happen is that the translated proteins will interact with this MOX and GANAP and this GPI-AP also. However, it is still in the immature form, or it has the misfolded structures. However, because of the absence of this Calnexin calreticulum cycle, the retention time within the cells get reduced, and this bulk of this misfolded GPI-APs will be expressed on the plasma membrane, either directly or through this P24 cargo. And when when the misfolded GPI-APs, and this third condition is about when the misfolded GPI-APs are present within the wild type cells, how they and if they are not able to acquire the native structure even after remaining within the wild type cells for a longer period of time, then what will happen? It will continue to recycle back in the Calnexin and calreticulum cycle. The rest of the process are similar. However, after some time, it will develop a acute ER stress, and this acute ER stress force these misfolded proteins to carry over through this P24 cargo and then expressed on the cell surface. And these things is not 100%, as some of the misfolded structures will also be diverted towards this ERAD, that is endoplasmic reticulum associated degradations. So however, that is not mentioned in this figure, but that is the general phenomena when the ER stress is introduced, that is misfolded structures are being directed towards the ERAD. However, because of the loss of the quality controls, some of the misfolded proteins will also carry over through this P24 cargo and expressed on the plasma membrane. And in this case, the paper talks about these GPI-APs. So we will discuss in our subsequent videos how they have come to this kind of conclusions and what are the hypotheses that they have used from the experimental results that they have presented in the paper. And meanwhile, if you want to read out this paper so that you can understand the videos in a better manner, you can go to the reference that has been provided in the description. That's all for this video. Thank you for your time.